Hello, I'm David Field, and today I'm going to talk to you about pre-processing of fMRI time series and detecting activation in processed data using the FSL software. Specifically, we'll be talking about using the FEAT tool within FSL to do this. The image I have on this slide is an image to indicate the effects of one of the most common kinds of pre-processing that's applied to functional images, which is spatial smoothing. More about that later. We will begin today by talking about some of the pre-processing steps, and then we'll switch to talk about how you detect activation in fMRI data before returning to a couple more pre-processing steps. And the reason I've put those at the end is because you can't really understand why you're doing them until you've understood the method that's used for detecting the activation in fMRI data. Okay, so when you launch the FEAT tool to process the data from a single participant doing what's called a first level analysis, you'll see this tab. One of the tabs is the pre-stats tab, which has all these options on it. So today we're going to talk about some of those, but not others. The three I've just crossed out with black lines are useful things, but they're not things that we'll be covering today. At least two of them are not, strictly speaking, pre-processing steps. So B0 unwarping, just briefly I'll tell you about that. That's basically something you can use if you've acquired what's called a field map in the scanner. The field map is an image which is an image acquired with the participant in the scanner that shows how the presence of the participant in the scanner is distorting the magnetic field and how that distortion varies across space. And those distortions will be happening in the functional images, and they don't happen so much in the structural images. So the reason you might want to do B0 unwarping is to use this map of the distortions in the functional images to adjust the shape of your functional time series prior to registration to the high resolution anatomical. So by unwarping it to remove the distortions that the person themselves created in the magnetic field, which were reflected in the functional images, you make those functional images a bit closer in shape to the anatomical before you do registration to the anatomical and then on to the template. So it's a useful thing to do if you've got a field map. Now, for fusion subtraction is something you would use if you were doing arterial spin labelling functional MRI rather than echoplanar imaging functional MRI. It's a less common form functional imaging. I won't talk about it further today. Melodic ICA data exploration. ICA stands for independent component analysis. So what that will do is it will just, without any model, it will just kind of find spatial and temporal structure in your data, and it will produce a long series of components where each component has a spatial map and a time course to go with that spatial map that will show the time course with which the activation in the spatial map is fluctuating. So it's quite good for identifying where there is sort of artifact activation due to motion, and it can also identify the actual activation from your experiment, and you'll know when it's done that because the time course of the component will look like the time course of your experiment, and the activation will be in sensible parts of the brain rather than all around the edges like the motion stuff usually is. So it's something you can do, and you can then subtract the noise components from your raw data as a form of pre-processing, but it's very time consuming. You have to do this by eye almost, so it's not part of the standard pre-processing pipeline, and we won't go into it in any more detail today. If you want, you can try it out on some data in one of the workshops or in your own time. So here are two pre-processing steps that I've put big red stop signs through, because basically there's never really a good reason to do these. These are just here for historical reasons. It's things people used to do years ago, and so I guess they're there in the options still, in case you want to replicate an old analysis from an old publication. So intensity normalization, that would be the process of calculating a transformation such that every single functional image in the data has the same mean intensity if you average across all the voxels in image 1, and that will have the same image intensity as an average of all the voxels in image 2 and image 3, etc. And one of the reasons that's a bad idea is because actually there can be meaningful fluctuations in overall average image intensity, which are due to functional activation that you're actually trying to detect. So you don't want to squash those differences due to variation in the bold signal by forcing every image to have the same mean intensity across voxels. A low-pass filter is a pre-processing step that's been superseded by something else called pre-whitening, which I'll be talking about later on. So a low-pass filter is not generally a good idea for fMRI. What it does is the low-pass filter removes high frequencies above a certain cutoff and leaves lower frequencies in the data, but we don't want to do that. So today we will be talking about motion correction in more detail than we did in the first lecture. We're talking about something called slice timing correction in detail. We will revisit BET brain extraction that we talked about in detail, that we talked about slightly in the first lecture, and we'll talk about high-pass filters. So those are all the things we will actually talk about from the pre-processing or pre-stats tab in FEAT. So let's begin with a bit more about BET, the tool for removing the skull and cerebrospinal fluid from the image in order to allow us to concentrate on just processing the voxels in the brain, and also to help the registration process. Now, this is really just to clear up some confusion that easily arises. Ticking this box, the BET brain extraction box on the pre-stats tab in FEAT, will cause BET to be applied to the functional time series. So the skull will be stripped from the functional time series by ticking that option. It won't cause FEAT to strip the skull from the anatomical image of the participant. You need to do that manually in the way we did in the workshop last week before you go in and set up FEAT. So a couple more brief notes. So these are actually two options on the miscellaneous tab rather than the pre-stats tab. If you tick the balloon help box, then that means that whenever you hover your mouse over one of the options, you'll get some help text appearing in a box, and usually that help text is very good, but after a while it can get annoying that this help text box keeps popping up, so then you can turn it off there if you want. Progress Watcher is the box you tick if you want FEAT to open a web browser showing progress of the processing, updating as different steps are performed. If you don't want to see that, then untick that. So we'll begin by talking about motion correction using the FSL program McFlirt. And what this does is it aims to make sure that there is a consistent mapping between a particular voxel X, Y and Z position and an actual bit of anatomy. And it does it using the 6 degrees of freedom rigid body transformation that I spoke about in the last lecture. And it does it by registering every image to the series midpoint image. So what that means is that every image apart from the reference image in the middle of the series has been moved slightly, and the output from McFlirt is a plot of those movements. Now, there was more detail last week about why motion correction can never be a perfect solution to the problem of the participant moving in the scanner, so do bear that stuff in mind. Also, a quick mention is given here to something called prospective motion correction, which is when you actually have the scanner move the volume around in which it's acquiring the voxels. So in effect, the set of voxels you're acquiring is moved to track actual motions of the head in the scanner in real time. Of course, it's a bit crude because you're only acquiring an image every 2 or 3 seconds. Therefore, you can only move your volume every 2 or 3 seconds. And so if the person's kind of moving with a high temporal frequency, it's going to be a bit problematic. But if you do choose this option, it's called PACE, P-A-C-E, in the Siemens scanner here at Reading, then you won't see much in terms of output from your post hoc motion correction with FLIRT. But that doesn't mean they didn't move. If you used PACE, you need to try and get into the Singo, the operating system for the scanner, to get out those values that tell you how the acquired volume was moved around during scanning, so that you can actually get some idea how your participant moved, because PACE can give you the false impression that they didn't move. So this is what the output from McFlirt looks like. The top plot here is a plot of 3 of those 6 degrees of freedom in the rigid body transformation. It's the rotations. So the kind of blue line is the rotations in X, the green line is the rotations in Y, and the red line is the rotations in Z. So we can see the reference image would be the 60th one here, and we tend to see that the displacements relative to that are larger at the beginning and the end of the time series, and that's not always going to be true, but it tends to be the case that the person is further away from where they were in the midpoint of the scan, when they're further away in time from that point. And the reason it turns out like this on the graph is because these are absolute plots. So for say image 20 here, the actual 3 values are the absolute difference between the position of the image that was acquired here and the one that was acquired in the reference image. And the same is true for this second plot here, the plot of the 3 degrees of freedom that are the translations that were made in X, Y and Z of the image of the head in order to match it to the position of the central reference image. Now the bottom plot is actually the most useful one generally. It's the total displacement plot. So rather than now looking at the 6 degrees of freedom separately, we're just looking at a total motion of the head, ignoring the actual directions in which that occurred. And the first thing I always do when I get one of these plots is I just look at the range of values on the y-axis, because like most bits of statistical software, FSL likes to rescale the range on the y-axis to make any differences that are present highly visible on the graph. So that can make very small differences look big if you're not aware of the fact that the y-axis has got a tiny range on it. So the range on the y-axis in this case goes from 0 to 0.5 and the units are millimeters. That's a pretty good participant. If someone has on the total displacement plot values of less than 1, that's pretty good. If you see 3 millimeters as the maximum on the total displacement plot, you would start to worry about that participant as a rule of thumb. Now there are 2 lines on the total displacement plot. The blue line is labelled here as being the absolute line, and so it's calculated the same way as the lines in the 2 graphs above, i.e. for image 120 here, it's just the absolute difference in position compared with that image compared to image 60, the reference image, whereas the relative plot gives you different information. This one is telling you about how much they've moved on a particular image compared to the image before it in the series. So it's just kind of like how much have they moved over the last two images, basically. And that's actually really useful, as we'll see on the next slides. Okay, this slide just reiterates what I said on the last slide, but with a helpful extra graphic in the form of this vertical line indicating the middle volume of the time series. So displacement is calculated relative to the volume in the middle of the time series for the absolute plots, and relative displacement, the green line here, is head position at each time point relative to the previous time point. Absolute displacement, remember, is relative to the reference image. Now, I want to talk about the utility of the relative motion plot. What it does is it enables us to find locations in the time series, such as the one highlighted by the red arrow here, where the participant has undergone a particularly brief and large motion, what we might call a spike, if you like, in the relative motion plot. And the reason these are particularly problematic relates back to what I talked about last week, which is that when you move tissue boundaries through the magnetic field, so for example when you move that boundary between the air in your sinuses and your skull, or between the brain and the skull, when you move that boundary through the magnetic field, it actually causes distortions in the magnetic field to change. And the change in the distortions of the magnetic field changes the actual image intensities that are recorded for particular tissue types. So then you've got a double problem. You haven't just got an incorrect mapping between voxel XYZ coordinate and anatomy, you've also got a change in the value of image intensity that was recorded, which is irrecoverable. So if you use the relative motion plot to identify images where there have been such large motions over a short period of time, you can actually improve your modelling of the data by modelling out these spikes. If you look at the actual images, you'll probably see that they have strange image intensity values in FSL view. So then you can set up a regressor that basically has a value of 1 for all the images that you consider to be spikes, and a value of 0 elsewhere, and do not convolve that regressor with any hemodynamic response model, so don't convolve it with the gamma or double gamma options in feet. Just leave it as it is, because it's not supposed to be modelling a blood flow response, it's modelling an image artifact. And that approach can sort of effectively remove those scans from your data and therefore stop them contributing to sort of large error terms or perhaps falsely being interpreted as activation. There is a MATLAB program that I used to use that kind of automates this process called rickspike.m, and that was provided by Rick Henson. So if you look him up on Google, you might be able to get hold of that, or perhaps some similar software yourself. Okay, we've finished with motion correction now. We're going to move on to talk about slice timing correction. Now, to understand why we do such a thing, let's recall that each slice in a functional image is acquired separately. Most functional images are actually acquired as a set of axial slices. So that red line is indicating the position of an axial slice on this brain. Now, people normally interleave the acquisition of their slices. So after acquiring this slice at the top of the brain, the next slice which is acquired is not the one which is going to be next to it in the final image, it's one further down. And the reason they do that is to avoid something called spin history effects from causing the voxel intensity values in one slice to have some influence on those in the adjacent slice. So by allowing everything to sort of calm down in one slice before you come back somewhere near it, you avoid that, and you end up with a pattern of acquisition like this. So you move from the top to the bottom of the brain, then you move back up to the top again and kind of fill in the gaps. So that's an interleaved acquisition sequence. Now think that if you've got a TR of 3 seconds, then obviously one slice is getting acquired in a few hundred milliseconds basically, and if you do it interleaved, then the time difference between the acquisition of adjacent slices can actually be quite large. Can be, let's say in this case, 1.5 seconds for a TR of 3. But what you're actually interested in is functional brain areas which are often large enough to span 2 or more of your slices, and the underlying scientific model here assumes that that functional brain area will all be activating together in temporal synchrony for a stimulus that it's processing, but you are modeling, you're actually recording it at 2 different time points. So let's consider now why these different slice timings that could occur within the same brain region are a problem. So remember last week I made it clear that the same design matrix is fitted to every voxel time course in the brain, and a design matrix is a temporal model. And what it does is, it assumes that all the voxel values in a single volume were measured simultaneously. I didn't say anything last week, did I, about the time points having to be jittered or anything like that. We just assumed that for a particular functional volume, that was all acquired at the same time. In fact, what the model does is it assumes that all those voxel intensity values were acquired at the midpoint of the TR during which that image was acquired. But we've now seen that it's actually not all acquired instantaneously, it's acquired in a set of slices that occur over the 2 or 3 second TR. So we need to consider what the implications of this are, and to do this let's think about 2 voxels which are part of the same functional brain area, but they're located not in the same slice, in nearby slices. And in the graphs on the following slides, the TR is relatively short, it's 1.5 seconds, so that's 1.5 seconds to acquire a whole volume of the brain, 1 data point every 1.5 seconds per voxel, but we've got a 1 second gap between the acquisition time of the 2 voxels in the nearby slices, covering our functional brain area. On this graph, this blue line just represents the hemodynamic response that occurs in this brain region of interest when we present it with a brief stimulus that it is specialised for processing, and we have a time 0, which is when the stimulus is presented, and on the y-axis arbitrary units for the bold response. So if that was the real bold response, if we were to measure that somehow independently of fMRI, and then also at the same time to record fMRI, then in the first slice, the blue squares indicate the actual values we'd record if we sampled that underlying hemodynamic response every 1.5 seconds. And submitted for modelling are these two time courses for these two voxels. So, you know, we know that the actual response in the brain, both in terms of neural activity and blood flow response, was the same in both of these, but what's actually submitted to the modelling stage is going to be, for the blue voxel, a response that peaks at 6 seconds, whereas for the red voxel we're recording a response that peaks at 4.5 seconds. And we know that neither of those are in fact exactly true, and that the difference in the timing of the response that we're seeing in these graphs is just an artifact of slice timing. Now, what we do is we assume that all of the voxels in an image were acquired at the midpoint of the TR rather than at the start or the end. So therefore the model, your design matrix that is going to fit better for voxels that are in slices that were acquired in the middle of the TR compared to ones that were acquired at the beginning or the end of the TR, where there's going to be a temporal offset and the model just won't fit as well. This is obviously not a desirable state of affairs. Okay, so what can we do about the slice timing problem? Well, option 1 is something that you covered in the workshop last week. If you include temporal derivatives of your regressors of interest in your design matrix, then those temporal derivatives allow the full model fit to be shifted in time to fit voxels in slices acquired far away from the midpoint of the TR. So then you would really need to look at the parameter estimates that you get on your temporal derivatives, and then you'd have to wonder, do these parameter estimates on the time derivatives represent slice timing issues, or do they represent the other problem I talked about last week, which is that variations do occur in the timing of the hemodynamic response between different brain areas and between different classes of people. So you'd have a kind of ambiguous situation. You would be unable to properly interpret the meaning of the parameter estimates on the temporal derivatives in the model. You also then need to do F contrasts instead of T contrasts, because when you consider the effects of experimental conditions on brain activity, those are definitely partly being modelled by the parameter estimates if you use for time derivatives, if you use time derivatives to solve your slice timing problem. So then you can't just do T contrasts on your main regressors, you've got to do F contrasts that cover both the time derivatives and the main regressors. We'll talk about that later, and most people generally prefer running F contrasts if possible. So another option is just to design your experiment in such a way that the 1.5 second shifts in activation peaks that are caused by the slice timing problem are not a problem for you. So basically, if you have a block design with quite long blocks, for example 20 seconds, then it just won't make much difference, and your model fit won't be negatively affected by the slice timing problem, so you can just ignore it and not correct for it. But of course, the psychological studies that most of you are interested in require the kind of stimuli that can't be all put together into discrete blocks. They need to be intermingled for them to have kind of meaningful, to be meaningful psychologically. So most people use this third option, which is to use a pre-processing step in which you shift the data from different slices in time by small amounts, so that it's as if all the slices were acquired at once. And that's what your pre-processing option in FEAT does. It actually alters the data to simulate what would have happened if all the slices were acquired at once.
Of course, the issue is that you don't actually have real data points to use. If you want to pretend that your data was collected at a certain time, it was actually collected a second later, then you need to do something called interpolation to work out what you think the value of image intensity would have been if the data point had actually been acquired a second earlier. Now, this diagram attempts to illustrate the process. So in this graph you've got space on the y-axis, so each row is a slice in your image. Okay, so that's like the bottom slice, that's the top slice in the brain in this sort of example, and you've got time on the x-axis. So what this shows us is that for this set of slices, which the FSL general linear modelling stage assumes are all acquired at the same point in time, we see the actual time points of acquisition for slices 2, 3, 4 and 5 relative to time point 1 for a sequence in which the slices were acquired sequentially bottom to top rather than the interleaved example I've been talking about so far. And the points in time where the pink shapes occur are actually the midpoints of the TR. Okay, so if it so happens that a slice was occurred at the midpoint of the TR, then no interpolation is required. If it was acquired at here, you've got this small interpolation to make. If it was acquired here, you've got a bigger interpolation to make. The interpolation is obviously going to be based on looking at the value in the next TR compared to the one you actually want to do the interpolation to, and seeing how much the image intensity has changed between the two TRs in order to sort of get some idea of what intensity it was likely to have in between. And this may also take into account multiple time points in order to try and sort of reduce the noise of the estimate. So also down here on the bottom graph we show the same thing for an interleaved acquisition, where the first slice was at this height in the brain, then the second slice was up here, third slice up here, fourth here, and fifth here, and here are the interpolation distances again in time. So that's all very well. But there are some caveats to this process. The interpolation, as you might have guessed from that diagram, is going to work quite well for relatively short TR, but once you've got a long TR, such as a 3-second or 4-second gap between this type between acquisitions of volumes, then there's just a lot of, it gets a bit more like guesswork, doesn't it? So basically the problem with this solution is it works well when the slice timing issues are minor, but it doesn't work very well when the slice timing issues are large and problematic. And there is a debate about whether to do slice timing correction before or after motion correction. FSL by default does motion correction first. Some people just prefer not to use this pre-processing step, but I think generally if you have an event-related design, you probably should use it, but be very careful to check with the scanner technician what order your slices were acquired in. If they were acquired in a non-standard order, then you just need to set up a little text file with the order in it, and FSL can read that in. Okay, so the next topic I want to consider under pre-processing is temporal filtering. That's filtering your data in time, and this is a long established method in any signal detection process for cleaning up the signal relative to the noise. So the signal we're trying to detect is the bold signal, the fluctuations in the image intensity levels that are related to our experimental stimuli, and therefore related to the time course of our experimental stimuli. So the idea is that the signal and the noise, that is the signal of no interest, which is in effect random or at least not systematic with respect to our design, they are present at separate frequencies in the data. And so if we can find a way of removing signal at certain frequencies, then we can attenuate or reduce the amplitude of the noise frequencies and thereby increase the ratio of signal of interest to noise in the data, which will make the signal easier to detect. Now, another way to think about this is in terms of oscillations or sine waves. So think about your experimental design as if it were something that oscillated, or a periodic thing, a sine wave. And if it's a kind of simple experimental design, we'll have a clear repeating pattern in time. Now, other sources of variation in the bold signal might vary more rapidly than that, that is, be a higher frequency sine wave, or they might vary much more slowly, that is, repeat much more slowly than the ups and downs of your experimental time course, and so those would be low frequency signals. So let's try to illustrate the different frequencies of signal present in the data a little bit. In this graph, the x-axis is just time, or perhaps TR, so effectively time, and the y-axis is the arbitrary units of image intensity from the MRI scanner. And across the top, I'm just showing a block design. So blue A is auditory, red V is visual, green R is rest, and then that repeats itself. So I've got a repetitive experimental or signal of interest. But you can also very clearly, so in a sense we can decompose that bold time course and the previous slide into these kind of three simplified sources of signal. Here's the high frequency signal, which is not of interest to us and is some sort of noise. Here's the low frequency signal, again not of interest to us and some sort of noise. And this shape here represents the frequency of the signals that would be related to visual stimulation in the experiment. So what we want to do with our temporal filtering is to definitely remove these low frequency drifts from the data, and we'll discuss the issues surrounding trying to remove these high frequency signals from the data as well. So before we move on, let's consider what causes the fluctuations in image intensity that are not caused by your experiment, which are just what we think of as noise. So the low frequency ones can sometimes have physiological causes, such as the participant gradually becoming sleepy over the course of a long experiment, or perhaps digesting a meal. But low frequency noise can also arise from the physics of the MRI scanner, and one way that can happen, if we think about a process called shimming, which is something we do once we've put our participant's head into the middle of the magnetic field, we shim in order to optimize the homogeneity of that magnetic field. But once you've done that, a slow drift away from that optimization occurs over time, and that in turn influences the resonant frequency of the hydrogen atoms. If you think back to lecture 1, you'll remember why that's important. And so you can get slight changes in image intensity, or even quite large ones, that happen over the course of maybe half an hour, like the ones perhaps I was showing in the last slide. Now the rapid oscillations, the high frequency noise, could come from the fact that the blood supply that you're actually trying to measure something about here is pulsatile. It's going to have a strong heartbeat component in it. But again, the physics can also cause high frequency noise. One source of that is what's called thermal noise, which you can look up if you want to understand in detail, but it produces more noise once the radio frequency coil is heated up, perhaps during a long scanning session. Okay, let's look at an example of a time series of data and the effects of temporal filtering on that time series. Now the example is not going to be a voxel time course of image intensity values from the MRI scanner. The example is going to be a time course from something called the human circulation balance, which I recently recreated. The human circulation balance was originally created by Angelo Mosso in Turin in Italy in the 1870s or 1880s, and it was actually one of the first non-invasive neuroimaging experiments, probably the first, and it's probably the first time that anyone got serious evidence to link blood flow with mental activity, although unfortunately the original manuscripts were lost until recently, and even when they were found and translated, it wasn't absolutely clear that Mosso had really proved that there is a link between blood flow and cognitive activity. And so actually before the information became available about the details of the original, I ran this thing as a student project with Laura Inman, and after a few iterations we got it to work. So I'll just tell you a bit more about this and use it to illustrate temporal filtering. So this is our apparatus, which doesn't look as nice as Angelo Mosso's original one, but kind of is the same basic principle. So we have a fulcrum here, basically a sharpened bit of steel with a V-shaped bit of iron, angle iron, resting on it to produce a kind of balance or seesaw, essentially a lever. And because it's balanced on a knife edge, it's very sensitive with low friction. We can put a participant on here with their head up at this end. Here's a set of scales, and because the person's resting on the scales with a little bit more weight on this end than there is on this side of the fulcrum, we register a net positive reading on these scales of a couple of 100 grams. We can then use this mirror and a computer to present them some visual stimuli and do either a block design or an event related design, just like people do in an MRI scanner. And the question we wanted to ask with this is whether we could detect a change in the total volume of blood in the brain that was related to information processing. So that's a different question from what fMRI measures, because fMRI is mainly sensitive to regional redistribution of blood flow from one part of the brain to another. And of course what you're directly measuring is the levels of oxygenated hemoglobin relative to deoxygenated hemoglobin, whereas this thing measures a value in newtons or grams, if you like, which is related to actual volume of blood, which is of course a different variable. So one of the experiments we did involved a brief stimulus being presented every 25 seconds. Could be an auditory stimulus or a visual stimulus or something that was both auditory and visual at the same time, and we wanted to then see what the response was to that, but also we wanted to allow plenty of time, for whatever response happened to return to its baseline. Because we didn't know what the shape of the response would be, we couldn't do the kind of thing they do in fMRI where they model signal summation. We just had to see what the shape of the response was. So this graph shows a raw data time series with up to 570 seconds of data collection on the x-axis and force on the y-axis, and you can see that there's lots of high frequency fluctuations in this data and maybe some other stuff going on as well, and we want to be able to detect something that's repeating every 25 seconds over about that period. So what we did first was a low pass filter, or rather a low pass temporal filter. So the low pass filter allows the high frequencies through and cuts off the low frequencies. Sorry, I've got that the wrong way round. The low pass filter lets the low frequencies through and cuts off high frequencies. And you can see the data already look a lot cleaner once you've done that. But it's still evident that in this data there's some sort of low frequency components as well, kind of curve here, and so we could use a high pass temporal filter to remove that, which allows frequencies above a certain cutoff through and removes frequencies below them. So after the both the low and high frequencies are removed from that data series, it looks like this. And you can see that it looks like something you could potentially model a signal in now. And the reason it's really important is because when you fit your model, it's going to account for certain percentage of variance in the data. But if you leave all that high frequency and low frequency stuff in the data, then the percentage of variance your model can account for is going to be low. And all that other variance is going to go into the error term. So when you come to do your t-test or other statistics, you're likely going to get a non-significant result. If you can reduce the error term by reducing the variance that your model can't account for, you're much more likely to see a significant result. And this is essentially the same thing whether you're talking about fMRI data or something else like this time series, or even something that's not related to the brain at all. If you want to read more about what happens in the experiment I was describing, then that's recently been published in Brain by myself and Laura Inman. So on this slide I'm going to talk about guidelines for setting the high pass filter for fMRI, although before I do that, let's just say what I forgot to say on the last couple of slides, which is that the sources of high frequency and low frequency noise in the human circulation balance were related to things such as the heartbeat, which produced the high frequency noise, the respiration cycle, basically as you breathe your diaphragm moves with respect to the fulcrum of the lever, and therefore you get quite a big signal in the data related to the participant breathing. So that's a slightly higher frequency signal but still lower frequency than the experimental signal. And then there are low frequency periodic signals in that data which we couldn't really identify the source of, but we were able to remove them anyway. So setting the high pass filter for fMRI. FSL recommend that the lowest setting for the high pass filter should be equal to the duration of a single cycle of the design. So if you have a block design of the type ABCA, BCA, BC, it would be equal to ABC. If you were to set shorter duration, such as the duration just equal to A or just equal to AB, that would remove signal from the raw data that was associated with the experiment, and that would obviously be a bad thing. If you set something much higher, like something equal to ABC, ABC, then you're not doing as well as you might, because certain amounts of low frequency noise is still going to be present in your data, at the point where you submit it to the general linear model. Now the difficult case is where you've got event-related designs that are a bit complicated, so you've got sort of 4 or 5 different types of event, and they don't happen in a kind of ABCDE structure. Let's say it's the A is less frequent than the others, and so you kind of don't get an A until you've had about another 15 stimuli. That creates a problem, and there is then a subjective element to setting of the high. So it's good to bear in mind the high-pass filter thing when you design your fMRI studies, and this is one reason why it's worth understanding the details of processing rather than just learning to click the buttons in the software. So if you had a design where 5 experimental conditions, they were blocks A, B, C, D, E, and it took 300 seconds to get through one cycle of A, B, C, D, E, you'd have to set your high-pass filter to 300, and that would allow a lot of the low-frequency fMRI noise to pass through to the modelling stage. You don't really want to be setting your high-pass filter much higher than about 160 if you can help it. Once you've got low-frequency noise in the data that you model, it's very easy for that noise to become correlated with the experimental time course, because the experimental time course is periodic like a sine wave, and often the noise is a bit periodic like a sine wave as well. So therefore they could easily become correlated in time, and the low frequency noise can then appear as a kind of fake activation related to your experimental time course. So not just in fMRI, but in any signal detection experiment, it's a good general principle to ensure that the signal you're interested in and the noise in the data are present at different temporal frequencies. Now, just a note, about subjectivity in setting the high-pass filter. For a while, with the SPM software, they would set it using a similar rule to the one I've described here, but then after a while they decided to just go for a default value of 128 for pretty much anything, which didn't make a lot of sense to me, but I think they were erring on the side of getting rid of low frequency stuff. Now, we saw in the example data from the human circulation balance that a low pass filter was very useful, because as well as removing oscillations with a longer cycle time than the experiment, you could use the low pass filter to remove oscillations with a higher cycle time than the experiment, i.e. that high frequency noise such as produced by heartbeat. Now, in theory that should enhance the signal and reduce the noise, and this is what people were doing in their analysis in the first few years after fMRI became popular as a technique. However, it's now been demonstrated that low pass filtering is not optimal for fMRI time series, because those time series have temporal structure within the sources of low temporal frequency noise. And what that means is that it's not random noise, it's noise which is predictable. i.e. it's not white noise. White noise is purely random. Because this low frequency noise has structure within it, it turns out that a low pass filter can end up enhancing this noise sometimes. And a better solution has been created to the low frequency signals of no interest in fMRI time series, using a measurement of something called temporal autocorrelation in the data. And the program that does that at FSL is called FILM, and what it does is it's called pre-whitening the noise, i.e. making the low frequency noise as if it were random. Now, this is something I'm going to return to and explain in a bit more detail later on, but this is also where I'm making a break with the traditional structure of fMRI introductory courses. By covering temporal autocorrelation and also spatial smoothing, after we've looked at the T and F contrasts that are used to detect activation, because the whole reason we need to do temporal autocorrelation correction and spatial smoothing actually relates to those T and F contrasts very directly. So once you've done the pre-processing steps that we've considered so far today, and once you've fitted the general linear model to your data, used the general linear model to fit your design matrix to your data, which was the topic we focused on last week, finding the parameter estimates for each explanatory variable to make the model fit each voxel time course, then you're finally in a position to start to answer the questions you actually set out to answer, which is often, where in the brain are the voxels that, using the example we focused on last week, prefer faces to houses, and also where in the brain are voxels that respond more strongly to houses than they do for faces. So as I said, we covered the model fitting last week. We set up a design matrix based on the time course of the face stimuli and the time course of the house stimuli, and we found the parameter estimates each voxel for each explanatory variable in the design matrix, maximizing the fit of the design matrix to each voxel time course. So how do we decide which voxels were activated in each experimental condition? For example, in the faces and houses experiment, how do we decide if a voxel was more activated when faces are shown than during the resting baseline? Well, I suppose a simple way to look at that is if the parameter estimate for the visual condition is greater than 0, then and positive, then the voxel was active to some extent in the faces condition. But we have to take into account the noise in the data, the residual variation. We can't take seriously any small difference from 0. This is the standard problem of statistical inference that you've learnt about in studying behavioural data. Another way to think about this is that we need to get some idea of how confident we could be that if we repeated the experiment many times, the parameter estimate would always be more than 0. Or more precisely, we test a null hypothesis, don't we? We test the null hypothesis that the actual parameter estimate in that voxel at that part of the brain for faces in the whole population is actually on average 0, and we calculate the probability that the parameter estimate we got in our sample, which is greater than 0, could have occurred just by random sampling effects from a population where the mean parameter estimate is 0 with some variation around it. That's our null hypothesis. So how do we actually take this noise into account and quantify our confidence that the parameter estimate is greater than 0, i.e. calculate a p-value? So we take the parameter estimate at a particular voxel and we divide it by the estimate of the residual or unmodelled variation at that voxel. So that voxel, if we consider the full time course, we have contributions to modelling that time course from all the different explanatory variables in the design matrix that have a non-zero parameter estimates, and that produces the full model fit, which you could go and take a look at in FSL view by loading in filtered func for one of the examples and then taking a look at the full model fits. Okay, so the full model fit is the time course, which is the best prediction we can make of the time course in a voxel given all our explanatory variables, and the residual variation is just a time point by time point comparison of the actual value of the data with the model prediction, and you calculate all the differences from the model, sum them, square them, etc. That gives you an estimate of the unmodelled variation, And this makes it clear to you why it's really important to reduce the size of that residual error term by judiciously choosing pre-processing steps and also modelling as much variation as you can in the data, because then your one parameter estimate for your one particular regressor that you're really interested in, for example the faces regressor, is going to be larger relative to that error term if you make the error term smaller, isn't it? So essentially what you've then got is a t-statistic which is converted into a p-value by taking into account the degrees of freedom, just as for behavioural data, SPSS, etc. So that p-value is the probability that a parameter estimate as large or larger than the observed parameter estimate would occur if the true value of the parameter estimate in the population at that voxel was 0. And the only actual variation present in the data was random. Now in FSL, t-stats are by default converted to z-stats, which simplifies interpretation because z can be converted to p directly without using degrees of freedom. Now another benefit of t or z stats rather than raw parameter estimates is that they standardise the fMRI signal across different voxels in the brain. Now why do we need to do that? You might remember from when you used FSL view to just sort of select various voxels in the brain and look at the time courses for functional data in different voxels, that some voxels had much higher overall image intensities than others, some showed more fluctuation than others, and even between different grey matter voxels you'll get quite a bit of variation depending on things like how far the voxel is from the receiver coil. Voxels which are further from the receiver coil, such as those in regions like superior colliculus or amygdala, are going to have inherently smaller fluctuations and therefore inherently smaller parameter estimates than voxels closer to the receiver coil, such as visual cortex or motor cortex voxels. So it therefore follows that if I want to compare the amount of activation for a particular experimental condition between two voxels, I can't really do it by just comparing the unstandardized parameter estimates. But because the overall amount of variation and therefore the overall amount of noise variation and probably therefore unmodelled or error variation tends to scale with the overall image intensity in a particular voxel, if we do this thing that we do when we calculate a t or z stat of dividing by the residual variation, then we do actually get a measure of effect size that's comparable across voxels, and we can start to then say, oh yeah, there was more activation in this region than that region, and of course we want to be able to do that, so it's really important that we can do this. Okay, on the last slide we saw that t and z are essentially metrics in which the effect size is expressed in units of residual variation, or units of standard error, if you like. Now let's look in a bit more detail at inclusion of what's called effects of no interest in the design matrix or model. You'll also hear these referred to as nuisance regressors. Examples of such nuisance regressors or nuisance explanatory variables include the head motion parameters. Now isn't that a bit odd that you first do a pre-processing step to try and adjust the actual images to remove problems due to head motion, and then you go and include them estimates of the head motions in the model as well? You'd think the problem should already be gone before the modelling stage, but I suppose people are reasoning that there is still some variation in image intensity in the data due to head motion because of the imperfections of the motion correction process. I mean, you can read literature about whether or not to include these head motion parameters. I think people disagree on whether it's useful or not. Another one is image intensity spikes, which I mentioned earlier, where you've got particular images with really strange effects because of rapid small motions of the head, for example, or scanner artifacts. Then there are physiological variables you can measure as a time course at the same time as your experiment is taking place in the scanner. So you can measure the galvanic skin response, the heart rate, and the pupil diameter, to give some examples. These are things that might actually vary due to your experimental stimuli, or perhaps just vary due to what happens when someone lies in a scanner for half an hour to an hour and gets sleepy. So those sort of variables, if included as regressors to model effects of no interest in the model, can sometimes be helpful. Or sometimes they could even be the effect of interest rather than the effect of no interest. They all act to reduce the size of the residual error term, and as we've seen, the t statistic is going to be bigger if you reduce the error term, and of course it's also bigger when the parameter estimate is bigger. A couple more points to make. Firstly, you can model the residual variation which is somehow systematic, such as that due to head motion, but some of the residual variation is truly random in nature, such as the high frequency noise caused by thermal noise. And so because it's random, it, by definition can't be modelled. The only time you would end up modelling it is if you were to overfit your data by setting up a design matrix that had a huge number of explanatory variables, and that these were set up in a very flexible way. For example, a Fourier basis set that includes lots of different frequencies, including some high frequencies, could model pretty much anything, including noise, but this is of course a meaningless thing to do, and if you do that you might find that you've used up so many degrees of freedom with your design matrix that you then kind of cease to get significant results anyway. Let's now talk about t-contrasts in a bit more detail. The first step in performing a t-contrast in FSL is to do something that has a fancy name but is actually very simple, called make a contrast of parameter estimates. And each contrast of parameter estimates you perform results in a new 3D volume being saved to the hard disk, where at each voxel location there's a value for the contrast of parameter estimates. So a cope is just a linear sum of parameter estimates. So we talked about some of those in the earlier slides, which were when we contrasted implicitly some of our individual parameter estimates with baseline to ask if a particular voxel was more active than baseline in a particular condition. So to represent that contrast with baseline as a cope, it's just the visual parameter estimate times 1 plus the auditory parameter estimate times 0, if we're using the examples that we worked with in last week's interactive spreadsheet for modelling voxel time courses. Or we could have the other one that we used in that spreadsheet, the visual parameter estimate times 0 plus the auditory parameter estimate times 1. Now, if we were interested in doing something that we didn't do in that spreadsheet, which was to explicitly compare the visual parameter estimate with the auditory one to try and find voxels that were more active in the visual condition than in the auditory one, we could do visual PE times 1 plus auditory PE times minus 1. And I think that makes a bit more sense to see that as a linear contrast of or a linear sum of parameter estimates, doesn't it? It's plus 1 times 1 and minus 1 times another. We could reverse that, visual PE times minus 1 plus auditory PE times 1, in order to see the voxels that are more active for the auditory stimulus than they are for the visual stimulus. Now, if there was a third condition in the experiment, say a tactile condition where people were being stroked with a feather or something, then we'd have a third term in all of these linear sums of parameter estimates. So we'd have there for the visual PE times 1 plus auditory PE times 0 plus tactile PE times 0 would be the thing that gave us all the voxels that were just more active in visual than baseline. Here, for identifying voxels that were more active in auditory than in visual, we'd also just add a plus tactile times 0 there in order to just stop that contributing to the particular contrast of parameter estimates for that case. Now, of course, because this is a simple, effectively a simple subtraction of the parameter estimates, it's not yet been rescaled in terms of the error variance to produce any t statistic. It's just a raw contrast of the unscaled parameter estimates. You might wonder why we bother with 1 minus 1. Isn't 1 0 enough for most purposes? Well, of course, if you did have a voxel that responded about equally to both visual and auditory stimulation, it would show up as active in both the 1 0 and the 0 1 contrast, and not be active in 1 minus 1 or minus 1 1. So the 1 minus 1 contrasts are useful for finding voxels that are selectively activated. But remember the caveat I talked about in week 1, which is that when you perform a 1 minus 1 contrast, don't fall into a trap of assuming that all the positive differences occur because a voxel is more active in the one, is more positively activated in the one condition than some smaller amount of positive activation in the minus 1 condition. A positive difference that you find can arise because both of them have got deactivation and one of them has got less deactivation than the other. So in order to produce the t statistic from the cope image, you then divide by what I talked about earlier, which is the value for residual variation at that particular voxel, the variation that could not be modelled by the full set of regressors in any way. So that is of course based on the raw time course values minus those predicted values from the full model fit. Now, what you see as red and yellow blobs superimposed on anatomical images that look so good and get fMRI into the media so often, those are produced by just generating a mapping between T or Z values at each voxel and colour or colour intensity. So all you're really doing is thresholding things at a particular number and then just mapping a colour onto that. So when you threshold an image in order to see just the areas that are most active in a particular contrast, you're just basically setting a value of T below which no colour is assigned, and you just see the anatomical image underneath. So for a visual minus auditory contrast, if you set the value of T quite high, such as 4, you would just see restricted activation in the visual cortex. But if you were to set it much lower, say set it at 1.5, you would actually start to see quite a bit of activation spreading out into the brain from the visual cortex, and also maybe some kind of random blobs appearing that might just be false positives. Right, this slide just shows you how contrasts of parameter estimates that result in T contrasts are specified in the FSL design matrix. So this design matrix just embodies the very simple design for let's say visual stimulation is explanatory variable 1, auditory stimulation is explanatory variable 2. You can very clearly see the time course of the different experimental stimuli. stimulus off, stimulus on, off, on for visual, etc. That red line there represents the size of the temporal derivative relative to the volumes. So each white bar here ticks off 10 volumes, so you can see that the TR is like about equivalent to 25-30 volumes in this particular experiment. So we've got the TR, the high pass filter. So C1 here is just called EV1, the visual one. So that's our 1-0 contrast. Voxels activated more by the visual stimulus than the rest condition. EV2, stimuli activated more by EV2 than rest. Here we've got the EV1 greater than EV2. Here we've got the EV2 greater than EV1. And here I've specified an extra contrast that people don't normally use, which is a 1-1 contrast. Sometimes people think about using it, but then they tend to realize that it's not a very good idea after a while, because what the 1-1 contrast will give you is a kind of average activation of the two explanatory variables here, and that average activation is going to be hard to interpret. You don't really want to see where we don't really want to see where the average of activation in response to vision and in response to auditory is significant, do we? That's not often a useful question. When we want to do that sort of thing, probably what we really want to do is an F contrast, which we'll look at on the next slide. So F contrasts can be used to find voxels that are active in any one or more of a set of T contrasts. So an F contrast has more than one row. So in this example we've got the visual stimulation, the auditory stimulation, and a third experimental condition as well, the tactile stimulation. So you can imagine what the design matrix would have looked like for that. If it was a simple one like on the previous slide, it would just have a third column in it, wouldn't it? A third explanatory variable. So what the F contrast does is it implements a logical OR between the different rows in the F contrast. So it doesn't do what we were doing with that 1-1 contrast in the last slide, a sort of average. It's not the average of X and Y, it's X or Y. So here our X is voxels that are more active in visual than in auditory, and our Y is voxels that are more active in visual than tactile. Right? So this would produce a map showing us where in the brain there's greater activation for vision than auditory or tactile. However, there's something I've missed out of the story, which is, and this is I think the thing that means people tend to shy away from using F contrasts in practice, is that the F contrasts are bidirectional. So if you put a 1 minus 1 here, it also implies the minus 1 1, the reverse. So the map would actually give us voxels where the activation was greater for vision than auditory, or greater for auditory than visual, or greater for visual than tactile, or greater for tactile than visual. And that's a problem because most scientific hypotheses are directional, aren't they? We don't generally want to predict that voxels will be either more or less active in the occipital lobe for visual than auditory. We want to predict that in the occipital lobe at the back of the brain they'll be more active. Now if you do find yourself running F contrasts and you've got this bidirectionality thing, you can then do various things such as employ a mask to make sure that the activation is only of one direction, or you could just have a look at the time course of the parameter estimates for particular time course of the fit of particular explanatory variables, or just to see whether it's modelling increases in the signal or decreases. So you can unpick it, but it just takes a bit of extra work if you're doing f-tests. Now, one situation where you really must consider doing f-contrasts is if what you've done is include temporal derivatives of the standard explanatory variables in the design matrix, and if you believe those temporal derivatives are really going to do something important, like for example you're relying on them to deal with the slice timing problem, perhaps because you've got some data where you aren't confident of the actual order in which the slices were acquired because you've got it from someone else's scanner and they've lost the file that tells you the actual order of acquisition, so you can't use the pre-processing option, so you end up having to use time derivatives to deal with that. Then you're really going to need to use an f-contrast where one row is going to be the contrast on the main explanatory variable, and then the row underneath is going to be the same contrast but performed on the corresponding time derivatives. And that's because, you know, in a case like that, the time derivatives really are modelling meaningful experimental variance, and so you need to see which, basically you need to then ask which bits of the model that are related to that particular experimental condition are really explaining variance in the data, and it's then both of them. It's both the standard regressor and the time derivative. It's common at this point in an introductory fMRI lecture to talk about the multiple comparisons problem, which is, as I may have mentioned in the first week, the problem for determining the actual p-value of the t statistic or z statistic at each voxel, given the need to take some sort of account of the fact that you're doing lots of comparisons, and that if you just use an uncorrected alpha of something like 0.05, you'll have a lot of false positives. Anyway, I shall be talking about this in detail in week 4 or 5, so I'm just going to skip this for now. Let's talk about temporal autocorrelation now. This is actually one of the things that people think of as pre-processing of the data, but I've put it here after the model fitting and t-test section because you don't really correct for temporal autocorrelation until you first fitted the model, as we will see. Now, the problem begins if you think about the fact that you need to know the number of independent observations at each voxel. Because this is a first level analysis of a time series, the observations are time points. So what we're talking about is the number of independent time points, i.e. number of volumes or number of TR. And the reason you want to know how many TR you've got or how many volumes is because when you calculate that t statistic, you need to, in order to calculate the error term, you need to divide the residuals by the square root of n, the number of observations, minus 1. And also you've got to need to know your degrees of freedom in order to convert t-stats to p-values. So you need to know what is the true number of independent observations in a time series. However, image intensity in a voxel at time x is partially predictable from the same voxel's image intensity at times x minus 1, x minus 2, x minus 3, etc. And that's even true in baseline scans where there's no experimental manipulation. And this is because the random low frequency fluctuations in image intensity have structure, periodic structure. And so this means that the consecutive observations are not statistically independent of each other, and so the number of images is not actually equal to the number of independent time points in the data. The number of independent time points is actually some lower number than the number of images in the time series. And if you did just use the number of time points in your voxel time course as the n for the calculation of the error term, then basically you'd produce p-values that were too small, overestimate the significance of your data, and increase your false positive rate. The beginning of the solution to the autocorrelation problem in fMRI time series is to measure the autocorrelation. You don't measure it in the raw data, you measure it where it's most evident, which is in the residuals. So after the full model fit has been produced, you could on a time point by time point basis subtract the value of the full model fit from the actual data point. That gives you a time series of residuals. Now this is why we can't do autocorrelation correction before we fit the model. We have to first fit the model in order to get the residuals, right? So that's why one of the reasons I'm covering this after the other pre-processing steps and after model fitting. So if that's our original time course of residuals, what we do to assess autocorrelation is compute the correlation of a voxel time course with itself shifted in time. So if that's the original time course of residuals, then we can shift that time course by one volume in time and calculate the correlation between original and the new time course, and then we shift it by two volumes in time and once again calculate a correlation with the original time course. Now if you were to do this in SPSS, you'd set up something like this. Here's your volume numbers down here, the original time course of image intensities. Of course, if you just correlate it with itself here, it will have a correlation of 1, shifted by one volume, because we now don't have a data point to correlate with volume 1, but you can see this is all shifted down by 1. This is shifted down, the next column is shifted down by 2, etc. Now once you've got these correlations, you can plot the degree of correlation against the degree of shift. So here is an autocorrelation function of residuals for an fMRI time series. R is a correlation coefficient, so varies between 1 and -1. Lag is just in TR here, so it's a lag of 0 obviously gives you this perfect correlation of 1. You see that for lags of 1 and 2 there are kind of significant correlations above 0, positive correlations. We also see some correlations at much longer lags here which are negative, and I suspect that that's because in this time series the high pass filter has not been applied. If the high pass filter has been applied, you shouldn't really see those, I think. What we're most concerned with is those correlations at lag 1 and 2. So that orange line was just to indicate what it would look like if the temporal structure of the noise in fMRI data was white, i.e. there was no structure in it, it was random, which is what the statistics want to assume. The autocorrelation structure would be such that there'd be a perfect correlation at lag 0 and then 0 correlation at other lags. And that just highlights the two problematic lags. So, one strategy regarding temporal autocorrelation that was sometimes employed in the earlier days of fMRI is to avoid it. Because the value of a voxel is more or less predictable from nearby time points about 3 to 6 seconds in the past, if you use a very long TR like 6 seconds or 7 seconds, you mostly avoid the problem, and your time points would have sufficient independence from each other for the assumptions of the t-tests to hold up just by basing n on the number of volumes you'd acquired. As I said on the previous slide, you can also get predictability of the current time point from more distant time points, but the high pass filter should really deal with that. Now the option that FSL gives you for dealing with autocorrelation is called film pre-whitening. So what you do in order to implement film is you first fit the model, including the regressors of interest and of no interest, to the voxel time course in the way that we've been seeing so far in this course, and you just fit it ignoring the fact that there's also correlation in the data. That enables you to get the residuals. You use the residuals to estimate the autocorrelation structure, which is that graph I showed you in the 2 slides ago, and then that estimated structure can be inverted and used as a temporal filter to undo the autocorrelation structure that's present in the original data. So if you then ran the test for autocorrelation again, you'd end up producing a graph where, like the orange line, showing that there was not any longer any autocorrelation in the data. So that means your time points are now independent, and the number of time points is now truly the degrees of freedom. And you also apply this filter, which is created by inverting the autocorrelation structure, to the design matrix. And if you want to see why you would do that, you should look up the matched filters theorem. Basically, if you're conditioning the data in a particular way by applying a filter to it, then if you apply the same shaped filter to the model, you will almost by definition cause the model to be a better fit. And so FSL does this both for the high pass filter. It by default will apply the high pass filter to the model as well as to the data, and it will apply the autocorrelation filter to the model as well as to the data. So after all this has been done, the general linear model is fit again on the adjusted data, and then you can run valid T and F tests. Now to select this film option, you tick the pre-whitening box on the stats tab in feet. In the past this was considered very computationally intensive, which was another reason people didn't want to do it, but nowadays PCs are fast enough to make it manageable, and there are almost no circumstances where you would turn this option off. The final topic for today is spatial smoothing. Now fMRI noise doesn't just vary across time, it also varies across space, and spatial smoothing is a way of reducing that spatial noise and thereby increasing the ratio of signal in your data to noise. And I'm covering it at the end because spatial smoothing is something that's particularly relevant to how well your statistical model is going to fit. Now, spatial noise in fMRI is less problematic than the temporal noise we've just been talking about, because if we look at the spatial autocorrelation structure of fMRI noise, it is a bit like that orange line that we saw on the temporal autocorrelation graph. It basically isn't any autocorrelation. It's essentially random, essentially independent from voxel to voxel, and the noise has a mean of about 0. And in that situation, if you average the image intensity across several voxels, those random fluctuations will tend to average towards 0. Whereas if there is any signal in a voxel that is perhaps common with the surrounding voxels as well, and that signal is related to the experiment, then that's a signal which is all positive or all negative, and so averaging it won't cause a tendency towards 0. It will leave it more or less unchanged, and because we've reduced the noise variation, we've therefore improved the ratio of the signal to the noise. Another important reason why you might consider smoothing your individual participant data is because it reduces the anatomical variations between participants, which are going to remain even after registration to the template. Basically, smoothing blurs the images, And so if you think about resulting activation maps, you might activate the same functional structure in each of a set of 10 participants, but although it's the same functional structure, individual differences in anatomical structure mean that the actual XYZ coordinates of the peak are somewhat different in each person, and perhaps even the blobs don't overlap for some of the individuals. So then if you smooth the individual data, because it will make the blobs a little bit larger for each person, you're actually going to get more of an overlap between the different individuals in the scenario I've just explained, and that will be beneficial when you come to do your group level analysis that we'll be covering next week, where you're essentially in a nutshell averaging across people to find where on average there's an activation. Okay, this is a slide that just explains exactly what you're specifying with the number that you enter in feet to specify the smoothing. What FSL asks you to specify is a Gaussian defined by its full width at half maximum. Now, so here on this graph the Gaussian is being shown in red. Here on the y-axis we indicate half its maximum value. And so if we follow across to the x-axis, and then we can see the point on the x-axis where the function attains half its maximum value, we can draw a line down, and then we can do the same on the other side of the Gaussian. So that gives us two values at which the Gaussian has a value which is half its maximum in terms of the x-axis, which is space, and then the difference between x1 and x2 is therefore the width of the Gaussian at half its maximum, and that's the number you're specifying in FSL. The number is that width. So the smoothing is actually broader than the width you give, as indicated by the rest of the Gaussian curve. So based on the number you give, FSL actually creates a three-dimensional Gaussian, and it moves this Gaussian around the image. It, if you like, convolves the image with this filter, and so if you move this image around the filter, so move this filter around the 3D image, then when it's centered on a particular voxel, what it will do is it will replace the value of that voxel with the average of itself plus those of the adjacent values that fall under the Gaussian. However, the average is obviously weighted by the y-axis value of the Gaussian at the appropriate distance of the contributing voxel, and so voxels that are further away contribute less. And note that this is all done based on the original values, so when you move the smoothing kernel on to the next value, you're not using the replaced smooth value to contribute its value to the new smoothing operation. You're using just the original values. Here are some examples of the effects of smoothing on EPI images. Top image, raw data, no smoothing. Applying a smoothing kernel with 4mm full width at half maximum does this to the image. You see it's blurred it a bit, and a 9mm smoothing kernel does this. You can see that the anatomical information that you can discern in an EPI image is much reduced by the time you've applied a 9mm smoothing kernel, although such smoothing kernels are not uncommon in the literature. Now, the reason people do smoothing is that it helps them to detect activation more easily. Here is a contrast that produces some activation, and in unsmoothed data that contrast produces this rather noisy map with lots of tiny blobs scattered around, whereas once we've smoothed the data and then run the same analysis on it, we get this much cleaner picture which is easier to interpret, isn't it? You've got to have less to say, you've got to have less odd things to talk about in your discussion section of your paper if you've got smooth data. However, sometimes people don't smooth their data. So let's think about when it's a good idea and when it's not. It's generally a good idea if you don't care much about voxel by voxel resolution, because obviously doing smoothing does lose you some of your original spatial resolution. If your original voxel size was 3 by 3 by 3 millimeters and then you apply a 9 millimeter smoothing kernel, your effective spatial resolution is now much less than you started with. But if you're not particularly concerned with finding clusters that are smaller than a handful of voxels in terms of your original voxel size, then smoothing is going to be fine. And the reason people often do it, of course, is because they need to improve their signal to noise ratio because they've got a relatively small amount of data to work with. Of course, if you were able to run hundreds of participants, then you wouldn't need to smooth individual subject data, because the averaging that would happen at the group level when you looked at the group result would be sufficient anyway. So if you're averaging results over a group where functional anatomy and organisation isn't precisely known, so you can't go in and define a priori regions of interest easily, then again smoothing can help you. There's also a reason to smooth the data related to the method of correcting for the multiple comparisons problem that is popular particularly with SPM users. That's the FWE, family-wise error option in SPM, or what's called the voxel option in FSL. Now obviously we're going to talk about this a lot more in week 4 or 5 when I specifically cover the multiple comparisons issue, but basically let's just say for now that to meet the assumptions of this procedure for correcting the multiple comparisons problem, you need fairly smooth data, and obviously if you apply a smoothing kernel you're making the data smoother. But there are alternative ways of thresholding an image. You don't need to use FWE as your method or voxel in FSL, so don't feel obliged to smooth your data. Now a small smoothing kernel or no smoothing would be more appropriate if you need voxel by voxel resolution to answer your question. Perhaps going to use a small region of interest and use feat query to extract signal in that region of interest. So that's when you believe that your activations of interest will only be a few voxels in size, such as the superior colliculus. Of course, another time might be if you were very confident your task will generate large amounts of signal relative to the noise in the data. So visual cortex tasks are often like that. So then why throw away your spatial resolution? If you care a lot about the single subject results, for example, you're going to do computational flattening of the individual participant anatomies and register the functional results to those so you can show the results on a flat map, then you might not want to smooth your data because you want to interpret the activations relative to the anatomical features of the individuals. So finally, if you're mainly interested in getting region of interest data from very specific structures that you've drawn with high resolution on single subjects, you probably wouldn't smooth the data. Remember, you were doing some drawing of masks in FSL view in week 1. If you are going to smooth, you have to decide the size of the kernel, and you could decide it based on the size of the voxels in your data. Remember, if you want small voxels, your TR will generally end up being longer. If you're prepared to accept the default 3 by 3 by 3 sort of voxel size, you could have a TR of 2 or 3. If you want to go for really big voxels like 6 by 6 by 6, you could speed up your TR. So that's why your functional voxel size might vary, but obviously if you begin with such huge voxels you probably wouldn't need to smooth very much. Another way to think about the smoothing kernel is to think about the brain structure size that you're interested in looking at. Obviously both considerations are actually important. Now, I mentioned the match filter theorem earlier, which is from the signal processing field, and that tells us that if we're trying to recover a signal like a brain activation in noisy data, and fMRI data is noisy, we can best recover that signal by smoothing the data with a kernel that's about the same size as the activation of interest. So if you're interested in the amygdala, you might be able to work out the size of it from an atlas and then use a smoothing kernel of that size. Of course, often we don't know how big our activations are going to be before we run our experiment, so then we wouldn't be able to use that criteria, or if we did, it would be somewhat post hoc. It would involve running the experimental analysis once just to find the activations, and then running the analysis of the data again with a different smoothing kernel. And we could do that, but by doing so we would be using what's called experimenter degrees of freedom, i.e. sort of not sticking to the original analysis plan, changing it post hoc, and there's lots of fuss in the literature at the moment about how these experimenter degrees of freedom inflate the false positive rate in neuroscience data, so perhaps we wouldn't want to engage in that. Of course, even if you do know what structure you're interested in, for example the hippocampus, you might not activate all of it, and so if you're only going to activate part of it, your filter width would then be suboptimal. So there's a lot of problems with kind of doing it in advance just based on anatomy, unless you're kind of used to running experiments on that particular brain region. Now, a default that a lot of people use is to set the full width half maximum of the spatial smoothing to the voxel size times 2, and that seems to work fairly well for most exploratory studies. Okay, that's the end of the lecture on preprocessing.